Fear conditioning versus fear extinction. If two stimuli are coupled, we learn to associate one with the other. Fear is learned during stressful experiences associated with emotional trauma and is influenced by individuals' genetic predisposition, as well as by the individual's prior exposure to environmental stressors that can cause stress sensitization of brain circuits. Some fears are crucial for survival. However, other fears that are learned and not forgotten may hypothetically progress to anxiety disorders. PTSD, panic attacks are all forms of learning known as fear conditioning. The amygdala is involved in remembering the various stimuli associated with a given fearful situation by increasing the efficiency of neurotransmission at glutamatergic synapses at the lateral amygdala. Sensory inputs about those stimuli comes in from the thalamus or sensory cortex. This input is then relayed to the central amygdala, where fear conditioning also improves the efficiency of neurotransmission at other glutamate synapses there. Both synapses are restructured and permanent learning is embedded into the circuits by NMDA receptors, triggering long-term potentiation and synaptic plasticity. So that subsequent input from the sensory cortex and thalamus is very efficiently processed to trigger the fear response as output from central amygdala. Input to the lateral amygdala is modulated by the VMPFC and the hippocampus. Now, if the VMPFC is unable to suppress the fear response at the level of amygdala, fear conditioning proceeds. And the hippocampus remembers the context of the fear conditioning, making sure fear is triggered when the fearful as well as all the associated stimuli are encountered. Most of the contemporary treatments for anxiety and fear act by suppressing the fear output from the amygdala. And therefore they are not cures, since the fundamental neuronal learning remains in place.